So again, at the bottom of the RMD file for this section, you can find the solution to exercise 2. So I've just reloaded the number of molecules to get rid of the correction we did before, but you don't have to do that. To do the part 1, so the capture efficiency plot. So the x-axis is just the column sums of the number of molecules. That will get you the total molecules per cell. And then col sums of UMIs that are greater than 1. This will get you the number of rows for number of genes for each cell where there's at least 1 UMI. So that's all that this does, is it turns all the counts into 1 or 0 for bigger than 0 or equal to 0. And then you can just plot some nice axis labels. For part 2, for the amplification rate, you just take the sum of all the reads and divide by the sum of all the molecules, and that will give you the average amplification rate across your data set. Any questions on this section before we move on? Right, so the question is what the expected amplification rate is. So for the data sets I worked on, I would say about 30 times is pretty average, but that was for around 300 million reads across few hundred cells. We've mostly seen in about that range. One of the other data sets I've looked at was up to 50 times, which was a bit high, but 30 times seems about typical, although very low coverage will be lower. Any other questions before we move on from this? Nope. And I'm going to pass it over to Davis to introduce the next section.
Cool, thanks Tallulah. So hi everyone, I'm Davis. So I'm a developer of the Scater package, which we're about to discuss, and I'm currently a postdoc with Oliver Stegler's group at EBI, so across the campus from these folks. So for the rest of what we're going to do, we're going to sort of base it around the Scater package, and briefly what Scater gives us is a bunch of tools that are helpful when we're doing these sort of low-level analyses of single-cell RNA-seq data, so doing the things that we need to do in order to get to more sophisticated analyses downstream. So what we want to do is visualize the data, QC the data, pre-process it in various ways, poke it, see what it looks like, and then filter out cells and genes that we don't want so that we have a nice, clean and tidy data set that's ready for statistical analyses focused on whatever our scientific questions of interest are. So what Scater features are automated computation of QC metrics, transcript quantification from read data with pseudo-alignment, so that is using Callisto or Salmon as tools to quantify expression from read data. And so Scater is quite closely integrated with those two particular tools. It gives us what I've described here as data format standardization. So what Scater gives us is a particular type of object to hold our data. And why this is useful is because it combines the expression data, so the raw counts or however else we've quantified our data to start with, along with cell metadata and gene metadata or transcript metadata, and keeps it all together so that we can be sort of secure as we go through an analysis that whenever we subset genes or cells, we just have to worry about the one object and all the relevant information comes with it appropriately. We don't have sort of multiple matrices or multiple data frames floating around that we have to try and keep track of simultaneously, which just increases the sort of mental overhead and chances of making mistakes. So that's a nice sort of way to structure an analysis. And then the other benefit is that when you have a standard type of object in R, then you can write methods that operate on those objects, and knowing what is going to be in the object allows the programmer to do things a lot more securely. And so what we can do and what we'll see is that once we have our data in one of these Scater objects called an SCESet, which I'll explain in a little bit, once we've got our data in that, then we can easily apply methods to plot the data in various ways to calculate your QC metrics and so on. The other nice feature about Scater is that it's a bioconductor package, and the type of object that we're using is closely aligned to other types of bioconductor packages. So a lot of the tools that have been developed previously for expression analysis and continue to be developed specifically for single-cell RNA-seq analysis are in R, in bioconductor. So sort of doing our QC and pre-processing of data in R is quite nice, because then we have our data ready to apply the clustering, differential expression, and other tools that are available out there. So to give some brief overview of the Scater workflow, we can imagine sort of starting at the top of this picture here. And so we can start maybe on the left with raw RNA-seq reads. So if we've sort of our bioinformatics core or whoever else, so the sequencing facility has just given us the raw RNA-seq reads, then we can run Callisto or Salmon from within R, if we so choose, to quantify expression from the reads, and then quite neatly read those results into one of these SCESet objects in R. So in just a couple of steps, we can have the data in R in a format that's ready to handle there. On the other hand, if we have a count matrix that's been supplied by whoever, whether that's from a published paper or from bioinformaticians who've prepared that for us, then we can use this function called new SCESet to build one of these objects from a count matrix and from whatever other cell metadata or gene or transcript metadata we have. And so that brings our expression data and that metadata into this SCESet object. So this combines what we call assay data, which is the expression data in our case. And the nice thing about this is that we can, in the one object, have many different transformations of a particular data set. So the only requirement is that we have a matrix of a particular size. So genes, number of genes as the rows, number of cells as the columns. And so typically we'll have something like counts, maybe counts per million, and say counts per million on a log scale. And we'll carry them around in our object together, because for different types of analyses, we'll want raw counts. For different types of things, especially visualizations and that sort of thing, we'll want to work on log transform data. So this assay data aspect of the SCESet object allows us to have data in many formats in a very consistent way.
Does the counts table column names have to match the ASCII files?
Not necessarily. I mean, once you have your data in R, then you can call things whatever you like. There's a strict requirement in SCESet when you build it that if you have, say, a data frame of cell metadata, and to start with, say, a count matrix, then it's very strict to say that the row names of this data frame absolutely are identical to the column names of the count matrix. And we're very strict about that to make sure we don't accidentally assign the wrong metadata to the wrong cell. And also the same goes for what we call feature data, so the transcript or gene metadata. So that is also a data frame that can be supplied. And again, the row names of that have to perfectly match the row names of the count matrix for exactly the same reasons. But once you've got your data in say an SCESet object, then you can rename the columns, rename the cells however you like, rename the genes however you like. And again, the nice thing about doing that once you're in the SCESet object is that you only have to do that once, and like every matrix of expression values are changed consistently, and so are the names of the cells in the cell metadata matrix or so on. So that's a sort of advantage of getting it in there in the first place, and then we can manipulate it however we like. So having sort of got all our data into an SCESet object, then typically the first step is to use this calculate QC metrics function, which, as the name suggests, calculates a whole lot of QC metrics, which we'll discuss subsequently. And we can use these along with a whole variety of plotting functions to guide our choices of QCing and filtering the features, so that's the genes or the transcripts, and QCing and filtering the cells. And then once we have we've weeded out the cells and the genes that we're not interested in analysing further, then we can conduct some simpler normalization approaches, which we'll discuss later in more detail again. We can use plotting functions and other methods to look at what explanatory variables seem to explain a large proportion or an outsized proportion of the variability for different genes. So that can be helpful for trying to gain an idea of what known factors seem to be contributing to the variability we observe in expression between cells or between genes. And having gone through all of that, we can arrive with a tidy, filtered and normalized SCESet object, and then that's ready for downstream analyses. So that will typically contain the filtered count matrix, or say transcripts per million, also log expression values, and so that means we can plug that data set into methods that need raw counts as input or log expression values as input, and make use of the other tools that many people have developed for analysing single cell data. So that's the overall picture. So this might have been clear from the way I've described things so far, or it may not be, but the fact of using bioconductor means that the sort of terminology that comes along with this. So in bioconductor terminology, which obviously existed well before the single cell era, we typically assay numerous features, and by that we usually mean, at least in this context, genes or transcripts, and we're sort of generally, I think, here going to talk about analyses at the gene level, which are certainly a lot more common than analyses at the transcript level, particularly for single cell data. And we talk about samples, so that's absolutely equivalent to cells in the context that we're thinking about. So I've talked about the SCESet class already and what that gives us and some of the advantages of using it. So briefly, technically, it inherits bioconductor's expression set class, which has been widely used for microarray and bulk RNA-seq analyses, and many packages use this for the reasons that I've outlined: the consistency of subsetting and ability to have metadata alongside your expression values in a consistent way. What an SCESet class requires, what it must have, although these can be quite minimal, is this slot or element called express, so EXPRS, which is a numeric matrix of expression values, and the rows are the features or genes and the columns are cells. Typically, because most of the tools set up for expression that use ExpressionSet or similar, we typically expect this to be log transformed data. So typically the expression element of an SCESet class will be log transformed data. And so if you start with a count matrix and use the new SCESet function, then that function will produce an object that has the raw counts still in there, but the expression slot will be log2 counts per million, basically, from that. If you start with transcripts per million, then the expression slot will be log2 transcripts per million, plus a small offset. The other thing that these objects have are this phenodata slot, and so that is cell metadata, so the phenotype data coming from the sort of the bioconductor terminology. And what it also has is this feature data, so that's information metadata about the genes or transcripts that we're using. So that could be biotype, GC content, chromosome, et cetera, whatever might be helpful or interesting for us. So yeah, there's a lot of stuff out there about ExpressionSet if you're interested in the finer details there. And that's it. That's the introduction to Scater. So we're going to have a coffee break now, and we'll come back after that to talk about actually doing QC of expression and so on. Thanks.